Dear all, we'll be learning about immobilization of biomolecules on biosensors today. As we are aware that biosensors is about the biomolecules that is being attached to a particular sensor. So when we say biomolecules, there are certain restrictions for them. It's not like any other chemical compound or any other metallic compound that is being imbibed on a sensor. As they are sensitive to certain pH change, ionic strength, temperature, etc., there are suitable ways how they have to be immobilized on them. So today basically we learn about how they are immobilized on the surface of the biosensors. Basically, in a biosensor, we use a small crystalline substance called as quartz crystal. So why again quartz crystal? So this quartz crystal will enable us to maintain or to measure the mass changes even in a nanogram range. So basically the biosensors, the biomolecules are very, very small, and whatever the change that happens in them, that is after the reaction. So when an antigen combines with an antibody, basically the mass of the antigen will change. That change of mass is always in a very, very small range, that is in a nanogram range. So only this crystal will able to notice the microgram or a nanogram change. So this is being placed over a microbalance. So this is called as a quartz crystal microbalance. And why again quartz is that this crystal will create a good bonding between the biomolecules and the substrate. So there is a quartz crystal. Over the quartz crystal is where the biomolecules are placed, and these are all in a microbalance. So these quartz crystals are basically piezoelectric substances. It is a very thin one. Only when the crystal is very thin, the changes in the mass can be detected very easily. That is, the nanogram or picogram even changes can be detected by them. So what all they use in the quartz crystal microbalance is a very thin sheet of quartz, and that will be plated with gold. So gold and platinum and silver, when I spoke about surface plasmon resonance also, I mentioned that these three metals are widely used in biosensor because of their refraction capacity. So same thing here. So this, even the surface plasmon resonance principle can be applied on them. The basic metals that is being used to coat are gold, platinum and silver. Over that there's a very thin sheet of quartz that is being affixed. So this can be used for measuring. So this is how the electrode will look in the front side. So there's a very good quartz crystal, and above that there is a gold electrode, and there's a back side where these electrodes are being interconnected. So the main application of them is the main absorption properties of the biomolecules onto the surface of the quartz. So I'm repeating that why they are using quartz is one reason is that for the surface plasmon resonance, that you have a gold plated thing and whatever the refracted light can be passed very easily to a quartz. The second main reason why we are using a quartz is that the biomolecules can be easily adsorbed on the surface of this quartz metal. That's the reason. So what are the application of this sensor or the quartz is that you can use for binding of a protein, you can use for polymers, you can use any biomaterial, you can use for lipid molecules, cells, or any other biomolecule can be attached to the surface of the sensor. So now when you look at, these are the basic receptors what we use in a biosensor. They are antibodies. Sometimes antigens will be used, virus as a whole can be used, or cell as a whole will be used. There may be some inhibitors like enzymes or ligands or receptors again. Lectins, that is some polysaccharide molecule, glycoprotein molecule, cell surface, and some receptor surface like again membrane or protein or cell. Nucleic acid can be kept, that is either as a DNA or any other protein. Hormone and vitamin can be also be binded on the surface of this metal. So this will enable us to go with a very good sensor in a very miniature scale with quartz as a background. So these are the molecules which has a good affinity for this quartz crystal. So what is the measuring capacity is that there will be a spontaneous excitation of the sensor with the AV voltage. So all this will be mounted on a semiconductor device. So there will be excitation of a current or voltage that is being given to this quartz metal. So yes, this equation tells that as what is a change in frequency and what is a change in mass. So del M is the mass change and del F is the frequency change. So basically, when you initiate or excite the sensor with an AC voltage, any frequency change. So the frequency change in the voltage will happen when there is a positive or a binding reaction between the receptor and the target molecule. So when there is a positive reaction that is being going on, what happens is the frequency changes. As the frequency changes, you can measure the mass change, which is directly proportional to it. So that's the principle behind this QCM. And the deposition of the thin film increases the oscillation and resonance frequency. So the molecules will be in some resonance frequency. So that is why a thin film of these metals are also used. The metals in the sense it may be a gold plated or it may be a platinum or a silver plated. And the molecules, that is a metal molecules or ions on the surface, will be in resonance, which can be easily detected. So during a immobilization, there will be a hydrophobic reaction, there may be an ionic reaction, there may be a covalent attachment of the molecule. So these are the different methods how they immobilize the molecules. It may be hydrophobic, that is certain molecules which are having the hydrophobicity can get adsorbed on the surface of the quartz. There may be ionic reaction, that is if you try to use a negative ions or positive ions, you can functionalize the quartz crystal and make them to interact. And by covalently, you can attach based upon some chemical surface group. So these are the three main methods used. So hydrophobic molecules are like mainly the protein molecules. Some of them, they have a hydrophobic nature. So in case if they don't have the hydrophobicity, you can have a ligand called as hydrophobic ligand, and you can make the molecules get attached on the surface. So this is the base matrix, which is nothing but the QCM, that is a quartz layer. Over that you have a gold or a silver layer, and then if in case your protein is a hydrophilic molecule, it may not be able to get attached to the hydrophobic substance. So you can use a ligand. Ligand is nothing but a short molecule which will enable the attachment of the protein molecule indirectly to the quartz material. So ligand is a small protein molecule, what you can say, which may be a hydrophobic in nature, which can be attached on the surface, and then your protein molecule can be attached. So so in case if your protein is hydrophilic and the hydrophobic molecule, there will be ionic interaction between them, and that can be attached to the quartz crystal. Ionic interaction is mainly based upon the positive and the negative reaction. And in case if your protein is again neutral charge, you can make them charged, or you can make them ionically positive or negative by adding certain chemical compounds to them. And then you can add to the crystal molecule. So this is a base here, which is in blue color. This is a base molecule. Here, that is SO3 molecule is acting as a ligand between the protein receptor and the substrate, right? So yes, in case if your molecule is not having a good ionic reaction, you can use some chemical substance which will allow the bonding of the protein molecule or antigen or a cell as a whole to the surface. So this is the second method. The first method, as I spoke, it is a hydrophobic immobilization. That is, the hydrophobic molecules get easily attached to the quartz crystal or adsorbed on the surface. So that can be enabled by a small ligand. The second method is an ionic interaction where the receptor, protein, virus, cell can be attached to the base with the help of a small chemical molecule. Next is non-covalent surface interactions. So, or covalent interactions, what I can say, where again your biomolecule can be attached on the surface with respect to some covalently bound molecules. So, some covalent interaction can be enabled with respect to the biomolecule. And there are different ways of covalent immobilization. So sometimes they do not absorb on the surface, they absorb very weakly, sometimes they absorb with improper orientation. So what we do for uh, to go ahead or to nullify all this problem is that the non-covalent interaction, that is sometimes the molecules will have a non-covalent interaction to the surface, so they can be made covalently interactive, so that these problems, that is if they are absorbed weakly, again you may get false positive or false negative, it may get absorbed with the improper orientation. So all this can be avoided with respect to the covalent immobilization. So yes, if the molecule is not able to covalently bind to the substrate, you can use some chemical compound. So here it is a double sulfur disulfide bridge that you can find, and the molecule is being immobilized in a particular matrix which will enable them to bind to the surface. So this is the third method. So the first method is a hydrophobic interaction, second is the ionic interaction, and the third method is the covalent immobilization, where if the molecule is unable to create a good bonding with the quartz metal, that is if the molecule is non-covalent, then you can make it a covalent bond by a disulfide bond and by immobilizing the biomolecule. It may be any biomolecule like enzyme, virus, protein, or anything that will be immobilized on a matrix and will be connected to the quartz metal with the disulfide bond. So this covalent interaction will enable them to get absorbed well and the proper orientation of the molecule, because the sites, that is the receptor sites, should be open for the biomolecule to come and bind. So that can be enabled very easily. So these are some of the disulfide bond, and it is being bound to gold for certain stability reasons. This is an example of that. And other immobilization procedures can be used with the help of some functional groups. So functional groups like amino groups, carboxy groups, aldehyde groups, thiol group, hydroxyl groups, all this can be used for a immobilization procedure. So this surface density of the ligand should be optimized, and low density molecule can be used, and high surface density molecules can also be used for this immobilization technique. So this is how this is a pictorial representation of how they go and bind covalently on the surface of the uh, quartz, that is a biosensor. So proteins are very sensitive, right? So you have, when we are attaching them to a certain chemical bonding or to certain thiol groups or something, you have to be very careful about what is the physical change or chemical change, what is happening, because both will affect the receptor site of the protein. If the receptor site is not uh, stable, then it may affect the bonding. And DNA molecules are more stable than proteins, and it is very easy to immobilize DNA than protein molecule. So we will see like what are the steps. So these are theoretically we have learned what are the interaction that happen and how they can be detected. So when it comes into action, that is when you are really making a biosensor, how this biomolecule can be immobilized on the surface. First thing is they will modify the gold electrode to create some functional group, and the biomolecules also should be attached with respect to covalent attachment on the surface, cleaning of the surface of the electrode, modification of the surface to create functional groups by either dipping, spin coating. So these are the practical way how do they do the proper sensor fabrication. So mainly these proteins and nucleic acid, when you are used, they have to be immobilized on a gold surface of the quartz crystal. That's what we were seeing all this. So, and proteins are very sensitive to pH change, temperature change, so that has to be taken care. And non-covalently, when the proteins are fixed, sometimes through physical absorption. But in case if you are trying a chemical absorption, again the receptor molecules or the recepting site should not be changed for the protein, and they should couple the receptor to the goal surface by our linker molecule. So when you look at this linker molecule, this is a surface which is positively charged, and they like this is a ligand that will enable the molecule to be attached. So initially, as their molecule may not get covalently attached directly, you use a ligand for them to attach, and yes, they will be present in a particular orientation. So the cleaning steps involved, there is a surface cleaning using a piranha solution. So this is a chemical solution that was enabled to clean so that there is some like oxide molecules on the surface, all that will be removed. Now the molecule that is a surface is being modified, and you can look at the modified surface which is having OH group, NH2 group, COH group and all that, and this will enable the covalent bonding with the surface and whatever the molecule. So this is a ligand molecule that we are using. It is a amine group, hydroxyl group or carboxyl group on the surface, and then the protein molecules are linked to it on the surface, or if it is a DNA molecule, directly to the ligand. So again, I repeat, when you are using a protein molecule, they are very sensitive to pH change, temperature change, and the ionic interaction. So you have to take care when a protein molecule is bonded. So when there is a change in the physical state of a protein, the bonding may not be specific. So you should take care for doing that. While DNA molecules are more stable, so that is how they will be immobilized on the surface. Uh, these are the general ways how they are immobilized. Protein molecules and single strand DNA, you can use blocking agents. So these are blocking agents that is being placed in between. The reason why they are placing the blocking agent is that any unwanted bonding, that is false negative or false positive, can be avoided with respect to this blocking agent. So you block the surface basically. So wherever the receptor molecule is not present, it's not present on the surface, you have a blocking agent. Again, that is being done by a chemical treatment. These are functional groups that have been created. So thiol group, acid group, sometimes amine groups, carboxyl group. These are all acting as functional groups. They will be synthetically made or sometimes extracted or through chemical reactions. They do that. And the surface of that quartz can be modified accordingly with respect to all this thiol, aldehyde, epoxy, biotin. This is a biological substance. So when you want the enzyme, like when you saw in the sandwich and ELISA. So in sandwich ELISA, what happens? Already an enzyme will be placed, and then the molecule, then you have a secondary enzyme or a secondary antibody for the detection, and that will be attached to some chromogenic substance. So in such cases, you can use a biotin surface, streptavidin again for the color formation. So all this is for the surface chemistry modification, and you have all this amine groups and carboxyl groups for doing this surface chemistry. And after immobilizing the molecule on the surface, these are the basic way how they look. Okay, so mainly this kind of immobilization. So when it is a DNA molecule, it is simple, right? So you keep a single stranded molecule. So when it is interacting with the counter reaction, so that is complementary strand will come and get attached. So the complementary strand get attached to the sensor, and now the mass of this will change, and the frequency that is of voltage that has been given will change, which will enable for the detection of the concentration of the molecule that has got attached. So this is a protein molecule again, right? You have a blocking in between, right? So whenever the reaction happens, if this is a positive reaction where the target antigen has come and bound to the protein molecule, and now there is a positive reaction, so the frequency will change. The mass of the protein that is being attached on the surface will change based upon the positive reaction. So these kind of reaction, when it is a positive, you can detect the exact concentration with respect to this interaction, and can be analyzed. So this will be directly connected to a detector, and through a semiconductor device, you can detect how much of positive reaction has really happened here. So now coming to here, as I told you, the protein molecules are little sensitive, and especially when it comes to enzyme, they are very, very specific to the reaction. So any change in pH or temperature or even chemical reaction, that will change the surface chemistry of the protein molecule. So these are basic immobilization methods that is especially involved for a protein or an enzyme molecule, right? So the basic what we are seeing all this while is adsorption procedure, where to any substrate that can be adsorbed directly or indirectly. Directly means the biomolecule is directly attached to it. Indirectly means through a ligand you can attach. If they are non-covalent in nature, you can have a covalent bonding with respect to some chemical reaction that is happening. Or in case if you feel the biomolecules will move away from the target or something, you can have a cross-linking. So this will increase the stability. So cross-linking is done by again biomolecules again using some catalyst and using some biological reagent. They make a cross-linking. Or in case if the protein or the enzyme is very, very sensitive, you can have a encapsulation. So the protein molecules will be encapsulated onto a semi-permeable membrane, which will allow only the substrate molecule to enter. So these protein molecule or enzyme molecule will be covered or protected. So this will again enable a better interaction. This is entrapment using a matrix and a fiber molecule. They entrap the protein molecule and the enzyme molecule on the surface. So this is an entrapment method where they are enabling with the help of the protein molecule. And these are the basic protein immobilization and enzyme immobilization techniques, which will enable the protein molecules to bind on the surface or a DNA molecules to bind on the surface. So I hope this class was useful to you and you could understand how sensitive is a biosensor and how best we can absorb or covalently bind the biomolecules on the surface. That's it for today, dear students. I'm ending my class here, and thank you for listening.